Thank you for the opportunity. Right, so yes, recently we have developed a number of new tools for Bioisoster mining. And I'd like to start with a sort of intro to the concept of isosteres. It's been sort of invented about 100 years ago now. Irving Langmuir proposed this idea that if you have a pair of molecules that share sort of covalent topology and electronic structure, they should have similar physical-chemical properties. And then a number of people sort of expanded that notion, and like Ellen Meyer started to look at biochemical properties and activities, and the concept gained use in medicinal chemistry. And if you look at some recent blockbuster drugs at the bottom here, PDE5 inhibitors, let's say, you can see that these molecules are basically isosteres. Some atoms are being moved inside this heterocycle ring system, and the two molecules share activity against the target, PDE5. Now, when this concept started to be used in medicinal chemistry, at some point people suggested that maybe the idea can be sort of broadened and generalized to actually include pairs of moieties or groups or linkers that can be replaced without affecting much the biological activity, but perhaps actually changing the other properties. So it became more general and in some ways more useful, but it's also because of the breadth, you cannot really perhaps enumerate easily the possibilities for isosteric replacement anymore if you, think of it in these broader terms. Let's say, if you look at this replacements for the dopamine catechol, there's all sorts of possibilities that people explore, and they're all active against one or another type of dopamine receptors. Even though other atoms that are added, the shape of the molecule is not necessarily the same as the progenitor, sort of, but they do share certain, if you will, pharmacophore, the combination of the aromatic system with some donors and acceptors. So given that expansion of bioisosteric space, there seems to be an opening for chemoinformatic techniques to be used to quickly come up with lists of possible replacements for a given group. How this can be done practically? Well, a couple of recent developments, relatively recent developments in chemoinformatics help. One is that now we have large bioactivity databases from which we can draw the information that could tell us that some things are isosteric. I mentioned in particular Campbell here, it's about 2 million compounds and millions of activities, but probably Novartis and other large companies have comparable in-house databases as well that could be exploited. On the other hand, recently fast maximum common substructure algorithms were developed, and using that, the algorithm that determines the so-called matched molecular pairs were developed. What is that, the MCS? Well, imagine, let's just look at an example of a couple of compounds, let's say glutamic acid and ibotenic acid. If you apply the MCS algorithm, it will tell you fairly obvious thing that there is this pair of identical portions in this molecule, sort of the alpha amino acid scaffold, and then there is a piece that is different, right? It's kind of obvious, but the great thing about the MCS algorithm is it can do it very quickly over a huge number of molecules. So if we combine this perception of common pieces of the molecule and different pieces of the molecule with the data on activity, so for this pair, for instance, the above ligands of glutamate receptor, we can conclude then that this two pieces are bioisosteric, so this carboxylic acid, ethylene, and the aromatic, well, heteroaromatic acidic group, they are bioisosteres, right? So that's basically how we derive our bioisoster database from Campbell. So we applied this sort of algorithm to the entire database, and that creates the database that currently contains on the order of 26,000 terminal groups and over 40,000 linker groups, and 500,000 pairs for the linkers, and over 1 million pairs for terminal groups. And then it's queried by basically giving it some moiety, and you will see this in a minute. Interactive demo inside the ICM chemist, or, well, in your case, focus environment. So you give it a group, and it gives you the list of potential replacements. One important thing is that we added to this basic search algorithm also an empiric score. When we looked at the raw list that come out of this kind of searches, we saw that we do find interesting and sort of expected, or interesting and expected pairs, but there is also some sort of background that may not be necessarily relevant. So we wanted to add a way to sort them so that the most relevant hits come out first. And the score combines a couple of terms that are so knowledge-based and then the more physicochemical terms. So the knowledge-based terms try to address the following issues in the database. Due to sort of historical bias, in any particular SAR series, almost every group will be replaced, let's say, with a methyl, and some other very common groups. So there is inherently always a fairly high frequency of certain groups that are just being tried so much that here and there they always pop up against any query group. So we weighed down by adding a term that would account for how frequently this particular our query group is replaced by a given return from the database over the overall frequency of that hit. So that would weigh down sort of in non-specific highly frequent groups. And then the other term reflects the fact that inherently again in the SAR series some positions are tolerant of replacements because they probably don't interact much with the receptor, so more or less anything that you place there would have comparable activity. But it doesn't mean that the groups are really isosteric, they just don't really affect binding much in this particular context. So to reflect that, we would also look at a third group in the same series and find other third groups for the given pair on a given scaffold that would have a much lower activity. So do the examples exist where replacing at the same position would produce much lower potency of the compound? If yes, then probably the pair are the true isosteres. If no, well, we don't really know. So that term accounts for this specificity.
I have a quick question.
Let's just look at how this looks interactively before I move to the 3D part, right? So
one last quick question.
So yes, practically the interface we have to these searches is well in the drop-down menu that you get in the chemical spreadsheet. So there is chemistry, and you can go to find a bioisoster for a group. Let's go to 2D first, and so all you need to do is essentially just select the group, let's say this carboxylate, and say, okay, there are some options here. it can either build also full molecules or just show the list of groups, and it can also enforce the type of attachment atom. So in this case, if I were to check this, then you know, if there are some carboxylates attached to something else, which is not very common, but they would be filtered out. So this is the hit list. It basically introduces first just the you know, the query that goes, you know, with all ones in the score bar, and then well, let's say the tetrazole ends up being second. The volume is slightly off because it's a little bigger, but otherwise it is you know, perceived as a match across the you know, the other terms, and then there are some you know, variants with carboxylate a little bit elongated. Again, we get, you know, some drop in one or another term as it gets bigger. Then we get, you know, some terms like, you know, sulfonate that is, you know, obviously there has more acceptors, for instance, here, right? So some reduction in the score, and phosphonates, and etc., etc., etc. So this is like heterocyclic replacement, phosphonate. That's how it looks, right? So the part is about finding bioisosterism in 3D database. You know, PDB is fairly large now. Is 130,000 structures, over 20,000 distinct ligands. The number of ligands is kind of still dwarfed, of course, as compared to, let's say, Campbell. But because the 3D structures actually show the molecules in their relevant bioactive conformations in the environment, it actually turns out that you can extract a lot of data, a comparable amount of data from PDB, even though there are fewer distinct ligands, and I will just explain that on a small example. Let's say we have a few compounds that bind the receptor, in this case PDE10. So if you are looking at them just in 2D, you know, let's say the algorithms like MCS wouldn't really give us a whole lot to chew on. I mean, let's say it probably would find, you know, the common nitro here and then suggest that these moieties are perhaps, you know, the benzimidazole and the different position as quinoline are isosteric. But once we put them in 3D context, so look at the 3D complexes, the structures of complexes, and superimpose them, then actually a completely different picture emerges when you look at the 3D alignment produced by essentially receptor binding. So the pieces that are isosteric are not necessarily those that are attached to identical groups. So for instance, in this group of 3 molecules, the 2 that share the nitro group are actually not really isosteric in any sense. The nitro groups stick to the different directions. Whereas this pair, in fact, aligns along this bond, you know, where the chlorine is attached and where the nitro is attached. So we could conclude from this superposition that actually these groups are isosteric. And similarly, if we look at 3D alignment of these 3 molecules, again, you know, just looking at 2D, you don't see much, but in 3D you can discover that, you know, the sub-pocket here is occupied by, you know, 3 groups, the chlorine, the methyl, and the nitro from the different scaffolds. So because we don't depend on, you know, 2D matching of the scaffolds, we actually can extract significantly more, you know, replacement information than if we just had the 2D data for a comparable number of ligands. So another sort of challenge to generate the database from 3D is that PDB, it just has a collection of individual structures that are not anyhow bunched up together according to the receptor, and certainly they're not aligned to superimpose the pocket. But Rubin's database at UCSD actually affords just that. So they systematically collected this pocket families for different targets, and so now it has almost 3,000 targets, pockets strong, about 16,000 ligands. Yes.
Max, a question for you.
Put this offline?
Sure.
Mm-hmm.
All right. So with the Pocketome in hand, then we put together an algorithm or a script that actually goes through it and looks at each entry and essentially identifies all the pairs of molecules and more specifically pairs of bonds that are close enough according to some angular and distance criteria, and then extract the corresponding pairs of groups that are attached to the rest of the molecule by this matching bonds. We also calculated for each group the variedness in order to again take care of issues with groups that are not truly isosteric because they're sort of in solution, right? So that produced a database of 10,000 groups and about 300,000 pairs. And similar to the 2D database I just showed, you query it and then you can do some drill downs. So once you have the list of groups, then you can go down to individual pairs of structures and then actually view them in 3D. And so let me again go back to ICM and we can perhaps view how this happens. It was constructed, and going back to the ICM or focus environment, here we can try to search for, let's say, isosteres for the nitro group in 3D now. So here it found some groups, and yeah, this is somewhat raw. I would sort it by number of pockets. Actually, is more meaningful way to view this. Right. So unlike the other search, since we have the 3D data, we did not, for the moment, apply this type of scoring as in the other case, and just rely mostly on the number of pockets where this substitution is observed. You can sort by that and then see the example. To find the examples, you can drill down, let's say for the chlorine to nitro replacement. I can double click and find some examples. So there's a number of pairs apparently in the androgen receptor. There are other cases as well. And then actually we can drill down further to see examples of how these replacements look in 3D, in fact. So,
Can I ask a question?
yes, sure. There. Right. So I was just showing that you can, by double-clicking, load the example and see the environment of the group to understand how this actually occurs. Why one could be replaced by another in 3D, and if you double-click on another, it should load the other example to this group here. So that's the 3D browser of the isosteres. I think that's what I actually have. If there are more questions,